Welcome. I'm delighted to introduce Dr. Andrew Persichetti. He's our newest faculty member at Jefferson and MRRI. And he is an expert in what he's going to talk about today, which is this layer specific approach to fMRI imaging. But before he came here, Andrew had got his doctorate at Emory University, worked with Daniel Bilks, and then he worked with Alex Martin at the NIH. And I think we're very, very lucky to have them here. We're very excited. One more thing before we start, I just want to remind everyone that we're going to try to privilege questions and comments from the younger crew here first. So us old folks will try not to jump in as much. So without further ado, thank you, Andrew.
Thank you. Cool. Thanks for the nice introduction, Laurel, and thank you all for being here. Yeah, younger folk, speak up. And also everybody, feel free to interrupt at any time while I'm talking. So this is the Shrier Family Topics and Rehabilitation Science Lecture, and it's made possible by a generous gift by Nancy and Marc Shrier. So we're grateful for their support. There's continuing medical education credits available for attending this. I'll just leave this on up here for half a second. Take a sip of water. Okay. And we have no financial interests to disclose. Okay. I'll try. Okay. So today I'm going to talk about the role of primary motor cortex in aspects of skilled movements. And I'm going to present work using cutting-edge layer fMRI methods. And since these methods are pretty new, I'm going to first give a brief description of how they work. And then I'm going to describe 2 experiments using layer fMRI. And then end with a discussion about how we might apply it to the neurorehab work that we do here at Moss. So I hope by the end of the talk, everybody here is able to answer these 3 questions. So learning and executing skilled movements requires a range of cognitive and motor processes, from selecting a goal, mentally rehearsing beforehand, and calibrating and refining body movements. And there's a large network of brain regions involved in these cognitive processes. I'd like to pause. I hope this is a very familiar figure to everybody. It's from a chapter co-authored by Dr. Aaron Wong. And the data I present today, I'm going to focus primarily on primary motor cortex, or M1. And so M1 is centrally involved in movement execution, but its role in the more cognitive processes of skilled movements is debatable. And in fact, the neuroimaging data on the cognitive roles of M1 is a mixed bag. So today I'm going to focus on 2 specific processes involved in learning and execution of movement: mental imagery of movement, and then in experiment 2 about motor sequence learning, repeating over and over again a movement to get good at it. And I'm going to use layer fMRI to probe the layer-specific functioning of M1 during these processes. So let me first explain how layer fMRI works by discussing, just in general, how conventional fMRI works. Conventional fMRI measures local changes in blood oxygenation levels in response to different types of tasks. And so, just to be clear, at the very fundamental level of what fMRI does, it doesn't measure neural firing directly. We're measuring blood-related properties as a proxy of the neural firing. And the basic logic of an fMRI experiment is localization. So you can have somebody do a task, in this case, move your fingers or tap them together, and you can ask, where in the brain is this happening? And then among other places, you should see a blob of activity lighting up in primary motor cortex in the hand region of M1. And this is great. And these basic localization types of fMRI experiments are wonderful. And we learn tons from them. But using BOLD to measure responses obfuscates the cortical layer circuitry. So cortex is made up of 6 cytoarchitectonically defined layers. And we know, mostly from animal experiments, the functional organization of these. So for example, in primary motor cortex can be kind of thought of a 2-compartment model, where cortical inputs and connectivity to the rest of the brain go into the superficial layers of cortex, and output to the body, to the thalamus and the body, are in the deeper layers. And to be clear, we can't get at the resolution with layer fMRI of these very specific layers, but we can get to this kind of compartment level description. So you'll hear me talking a lot about superficial and deep layers, not about layer 2, 3, and layer 5 and things like that.
Can I ask a
Yeah.
clarifying question real quick? So you take fMRI in layers, so how is it different from, I'm just confused how the layer fMRI is different from just like taking fMRI and just like slices.
Oh, well, layer fMRI, yeah. So that's taken in slices, and they're often millimeter slices. So you're taking the whole brain kind of, no, this thing doesn't work. You all see that? Like this thing could be a slice. If you're taking your slices anterior to posterior, you're going to get a bunch of these slices, and they're going to be, I don't know, 2 millimeters thick or something. That's quite different. So potentially, if those slices are small enough, if they're sub-millimeter, you could start to look at them and kind of see. You could look across layers, right? But, well, conventional fMRI typically isn't sub-millimeter. It can be, but there's an added wrinkle to this problem, to this issue. And it's that, again, we're measuring blood oxygenation changes. And those changes are dominated by these non-specific large draining veins. So as blood rushes in response to a task, it then rushes out of these large draining veins up and into these huge veins on the superficial, on the peel surface. So that oxygenation signal then is dominated by the superficial bias. So if you were to take a layer profile with BOLD, conventional BOLD, it's just going to look like this. You're going to get tons of activation at the superficial, and it will just ramp down because you don't have the specificity to get at these smaller cortical vessels. So to answer your question, with sub-millimeter voxels, that's part of the equation, but you need these layer fMRI measures to really get at these layer-specific changes. And so instead of looking at oxygenation changes, we're using a method called VASO that measures changes in cerebral blood volume. So as blood flows in, oxygenation changes, and also vessels dilate and contract. And that's what VASO is getting at. And by getting at it, we can look much more locally at these much smaller veins. So as the blood flows in, these veins expand. And that's what VASO is measuring. It's actually, fun fact, a negative contrast, because what it's measuring is the tissue displacement as these vessels dilate, the tissue displacement around them. But for our case, we make them positive, so they make more sense to us. And so when we measure the cerebral blood volume in response to a task, we can now get these layer-specific responses. We will get a double peak response that reflects these inputs and outputs. Yeah.
How does the technique deal with the layers are all convoluted, and which edge
Yeah.
to the slice. So how does that work?
Different ways. So if you have, it's actually, I took it out of this talk, but so if you have isotropic voxels that are small, sub-millimeter, 0.8 or smaller, really, it doesn't matter. It makes it a little bit more difficult because you need to orient yourself to whichever gyrus you want to look at and then figure out how to segment it. But once you know where you are, you just find the white matter and the cerebral spinal fluid on the surface, and you just kind of segment. But it does make it, convolution makes it hard, and certain regions are very hard to do this in. So M1, there's lots of great reasons to focus on it. One, the signal's really strong because it's up here. Also, it's the thickest part of the cortex. It's about 4 or 5 millimeters thick. And also, the way that it's positioned, this hand knob, we can actually leverage that, and we put our slices perpendicular to the hand knob, so that when we then zoom in on it, each slice is just like going perfectly down this the cortex. Yeah, so it makes life a lot easier. So I'm going to use these layer fMRI methods, VASO, to look at mental imagery of movement in this hand selective region of M1. And so I'm going to ask two questions in this experiment. The first is, what does it mean for imagined and executed actions to overlap in M1? First, with conventional fMRI, it's, again, a mixed bag of results. Some people report that when people imagine moving their hands, that primary motor cortex is active, and some say that it's not. So the question of, well, if we take it that it is active, how is it overlapping with the actual executed movement, right? When I imagine moving my hand, I don't have to move it. So they have to be dissociable at some level. And the second question is, how does imagining an action or mental rehearsal result in improved performance? So in this experiment, we had 3 conditions. We had people actually tapping their fingers. We had people keeping their hands still and just imagining that they're tapping their fingers. And we measured EMG to make sure that there was no movements. And as a control, we had people keep their hands still and just wiggle their toes. And we did this in a slow event related design. So they did this for 6 seconds, then they rested for 9 seconds, then did the other thing, so on and so on. And we counterbalance these in a way that we can look at the direct effects of somebody tapping their fingers, and we can also look at how tapping their fingers is affected by what preceded it. So if imagine tapping preceded tapping, how does that change the activation? And so we can make our predictions here. So first, as I told you, we should see a double peak response when somebody's tapping their finger in M1, reflecting this two compartment input-output model. And we predict that when we have people just imagine their hands while keeping them still, we're going to see activity in the superficial layers, but not in the deep layers. And the toes are included because as an active baseline, we shouldn't see responses in the hand selective region during toe wiggling, and we can compare it to our other conditions. So first, we need to functionally define the hand region. So the hand region of M1, it's pretty easy to find. It's called the hand knob because it has this kind of knobby structure. So we can go in there visually, find it, and then we go in here and just segment, kind of to John's question. We can go and we can find white matter and gray matter boundaries. They're more visible here. And just segment those two borders. And then we're going to use the direct responses to the tapping condition as the localizer. So we can look at this hand region and we can look across it and find what our expected double peak response. We can find it here. We used a really basic regression model so that we didn't have to visually choose where this thing was. The regression model found these double peak responses for us, and then we can draw our layer mask over this region of M1. And this layer mask is 20 layers, and we do this. It's a rather arbitrary thing. There's lots of discussion about how this doesn't fully matter. The point of using 20 layers is to get a good, nice layer profile that we can then identify and then probe. So this is just example data from one person. And I'll point out these data are non-independent, right? I've gone in and used this tapping to find the region that I want to look at. So the double peak is given. So now we can look at our conditions of interest. So first, this is just average. We had 11 participants in this experiment. This is their tapping data averaged across all people. And you see that across all people, we get a nice double peak response. Again, it's non-independent. We basically forced it to show this layer profile. Now we can look at our conditions of interest, which is mental imagery. And we see, as predicted, we see a significantly greater response in the superficial layers compared to our active baseline of toe wiggling, but not in the deep layers. And we quantified this by, again, using our functional localizer. We took the peak activation in the superficial and the deep layers respectively, and just averaged the points on either side of it together to get a bar graph. And we can run an ANOVA, and we see, as I just described, significantly greater response in the superficial layers and a significant interaction between the superficial and the deep layers. So the question of what does it mean for mental imagery to overlap with executed movements in M1? We found that in contrast to the double peak response that we found in when people are actually tapping, in mental imagery, you only get the superficial response. So it's reflecting more of a motor plan and not the actual execution of the movements. So even when you drill down into M1 and you can look across layers, you can see that they're dissociable in this region. So in the second part of this experiment, we wanted to ask a separate question. How are the neural responses to the executed hand movements modulated by the preceded stimulus? These are called repetition effects. And to do that, the data I'm going to show you, to be clear, are all responses to the tapping condition. We've now just taken the tapping condition and separated it based on what preceded it. So in black, it's when it's repeated, when they did tapping twice in a row. Red is when it's mental imagery, preceded by mental imagery, and green, the toe wiggling. And in both brain and behavior research, a common finding is that you find reduced responses when things are repeated. We call this, in fMRI work, it's commonly called repetition suppression. So if you look in a region, if you look in this hand region, you'll expect to see a reduced response when people repeat finger movements compared to when they do a finger movement following a completely different type of movement, in this case, toe wiggling. So this is the kind of thing we're looking for, these types of repetition effects. And then we can ask, well, what happens when tapping is preceded by the mental imagery?
Is that suppression effect because the blood flow is already kind of in that area, or is it actually like a change in the activity?
Oh no, it's a great question. Well, it's thought to reflect the neural activity. Yeah, there's there's a handful of models of why it could be the case. A common one is called sharpening, where when you first do, like with this, tapping your fingers, lots of neurons in the hand knob are responding, even noisy ones. But as you keep doing it, noisier neurons will drop out and refine the firing. But there's several models of this. So here are the layer profiles to these repetition effects. I'll show them to you again by averaging over the peaks. And so in the superficial layers, we see as predicted from basic repetition suppression, we get a significantly reduced response to repeated hand movements compared to when they're preceded by the toe wiggling. And we also see a significantly reduced response to tapping when it's preceded by mental imagery. And our working hypothesis here is that this repetition suppression in the superficial layer might reflect improved synchronization with other cortical areas involved in motor planning and learning. By contrast, and quite interestingly to us, we find the exact opposite pattern of responses in the deep layer. We see repetition enhancement in the deep layers. And we think this is really cool and pretty unexpected. And we think that this might reflect a boost in like the gain of the signal. So as you get this more efficient processing in the superficial layers, that might work as like a gain knob to your deep layers to result in a more robust signal getting sent out to the spinal cord. So imagining an action results in improved performance, potentially by sharpening cortical input, as reflected in the superficial layers here, and enhancing motor output here in the deep layers. And the next step in this line of work is to see if we can link these dynamics, these brain dynamics, to behavior. Yeah.
Learning, thinking that they're doing these tasks in a single session, right?
Yeah.
And you notice that there is this decrease over time.
Yeah.
But say they were like doing a task where they're performing like repeated sessions, and then you call them after a week and have them redo it. Would you expect a similar response that you see?
Yeah, that's cool. So like repetition suppression, you mean? Yeah. Actually, yes. But to be clear, there's no learning here. This is like the most simple tap. They're just tapping fingers. We're really just asking. There's not really a strong behavioral question here. We're asking just how the brain is responding to these really basic movements. So yeah, no learning here. Just very simple. What is cool?
That is interesting though, because it may be that there is some kind of plasticity / learning even within session, but it's at a fine grained scale. That is, people are a little faster. They make slightly slower errors. So
Absolutely.
that would be interesting too. Another new next direction is to link even within session.
I'm so glad you brought that up. That's what Experiment 2 does. But what's really cool is repetition suppression to lots of showing somebody the same picture, whatever, like show them a set of pictures, take them out of the scanner. A week later, put them back in the scanner, show them some old pictures that they've already seen and some new, and you'll get repetition suppression. So, and these effects, in the scanner, I can't remember how far out they've gone, but behaviorally, like with reaction times, they go out long periods of time. Yeah. And it's an interesting question of like how that relates to learning. Like it's that type of learning would be so fundamentally incremental, and that it's something happening, and it is quite interesting. To Laurel's point, it would be interesting to know about within session effects of learning. So in this second experiment, we wanted to look at motor sequence learning. And so typically, skill learning involves strengthening new memories through consolidation. So better performance after you've practiced, you come back, and now you're better at it. And a typical an example of this type of consolidation is after sleep, right? So you practice something, you fall asleep, you wake up, you're better at it. And you're better at it with that sleep interval than if you did it in the morning, stayed awake, and then practice it after the same amount of time. But the same concept of this consolidation may apply to short periods of rest while you're practicing in the same session, and it's termed rapid offline consolidation. So as you're practicing your piano scale, when you take a break in between each time you practice the scale, the consolidation is happening at these very small breaks. So to test this experimentally, people will get a button box with the four keys on it, and they're just asked to, they're given a sequence to learn. So that, for example, this sequence, and they'll do it over 20 seconds of practice and 20 seconds of rest over and over and over again. And you see across people a learning curve, right? There's this early learning period where they learn quite quickly this sequence, and then this kind of asymptotic period afterwards. What's been shown, what's really cool, is that this early skilled learning it can be explained by cumulative micro offline performance gains, right? So if you look at these, the total learning based on, you can ask about while they're actually practicing, what is the difference between when they started versus when they stopped practicing? Or you can ask, what's the difference between the offline when they first end a sequence, take a break, and then start the next one? And what people have seen is that the total learning, when you average across this whole early learning period, is explained more by the micro offline learning, but not so the breaks in between, but not during the actual online practice. So we took this paradigm and looked at, again, primary motor cortex to look for layer-specific changes during this online learning. So we had 13 participants doing this same paradigm. And in our 13 participants, we see this learning curve across all 13 people. And if we do the same analysis that I just described and look at online versus offline learning, we see the same thing, where the total learning is explained by the offline, but not the online learning. In our experiment, we added a second condition. So we call this the training sequence block. We added a second condition that everybody did afterwards, which is the same structure, except there's going to be no learning of the same sequence. Every block is a random sequence, right? So they're always doing a separate sequence each time. Same finger movements, same button presses, just always in a different order. So everything's basically the same except for the learning. And when you look across our 13 participants here, you see there's no curve. As we predicted, there's just kind of random changes from block to block. And I say random, but it's not totally true. It actually matters kind of what that sequence is that they're asked to press. Some are kind of easier or harder than others, but doesn't matter for our purposes. So that's our paradigm. We have the training task, and then we have a separate task where they're doing the same sequences, same buttons responses, but always random sequences. So we're going to look at primary motor, same region of primary motor cortex. So we need, again, an independent functional localizer. So in addition to the training and the random blocks, we also had people perform two separate runs where they're just asked to repeatedly press each button in turn. So in this one, they just see like the numbers 1 through 4, one at a time, and they just, for a couple of seconds, and so they just press like 1 1 1 1 1, 3 3 3 3 3, 2 2 2, like that over and over again for a while. And we do that so again we can look at M1 and find a double peak response. And again, we can draw our layers over our double peak response. This is again, this is the result of that localizer data averaged across 13 people. Again, it's non-independent, where you're basically forcing it to have this double peak response. We've defined it that way. I'm showing it to you here, though, to show you too that we define this on this whole separate data set. And now I'm just taking our training and random tasks and just averaging all of that together, just to show you that we have really nice alignment of the localizer, the region we defined as the localizer, and our tasks. So these peaks line up very nicely. So the first analysis we ran is a pretty basic analysis like the one I just described in the previous experiment, where you just average together activation from all of the blocks and see if there's differences between learning sequence and random sequence. And so here I'm showing you this while they're actually during the practice parts of the sequence, while they're actually pressing buttons. And we're looking at it training in green, random in purple. And we can see that there's differences here while they're actually pressing buttons in the superficial but not the deep layers. As I told you before, we set this up because we were interested in this rapid, this offline consolidation happening in between these blocks. So in addition to looking at the practice blocks, we looked at the rest blocks in between while they're practicing, and this is just all that averaged together. And we see differences in both the superficial and deep layers here. However, this doesn't tell us anything about how they're involved in learning, right? We've averaged across all of those that whole learning curve and just asked, is there differences, right? But again, we want to get at this change in the activation while across this learning curve. So we did this using a model. So there's two parts to this. The first part is a pretty simple we fit an exponential model of behavior for each person's learning. And so you can see pretty clearly here, like, so whatever, this guy, subject 2 on the top, has a fast learning curve here during the training. Compared to random, it's basically flat. And from our exponential model of behavior, we can get two parameters. We get the speed of their button presses during correct responses, and we can also get their learning rate. And so we're going to take this learning rate and the speed of correct sequences, and we're going to use a ridge regression to ask, to model the behavioral gains during the rest blocks across trials on the y-axis, and that's the learning curve average across, going down, and across our layers, and that's the double peaked response to the localizer there. And so what we see here, uncorrected, this is just p. If you just look at a p-value of 0.05 uncorrected, we get the model has fits in superficial and deep layers. Real quickly, just to explain the blue and the red. So blue is a negative correlation. So as less activation during rest means better performance over the trials, whereas the red would mean more activation during rest, you're getting better. Okay. And so after FDR correction of these model results, however, the only thing that survives are these negative betas in the superficial layers. So this is, this was the correlation with the learning rate parameter. We can also do this with speed. And again, when we FDR correct it, we see that the activation is happening in the superficial layers. And so what's pretty cool here is that, I think, is that these models are showing you, so the learning rate is negatively correlated with the activation during rest early on in the learning. As you're learning, as rest activation is suppressed, you're getting better. That flips and starts to turn into showing you this sustained speed change later on. When we look at this, when we apply these models to the on blocks, when people are actually practicing, we don't see anything significant during these practice blocks. So the model, it explains no variance in the learning curves. So we think that this correlation in the superficial layers, this negative correlation with rest, is consistent with a reduced or sharpened inputs to the superficial layers of M1 during rapid offline consolidation. So in these two experiments, I showed you two aspects of practicing and learning skilled movements using layer fMRI. So in experiment 1, we use mental imagery to kind of get at this question of how does mental rehearsal work, right? If you're going to take a free throw in basketball and you just imagine yourself doing it, why should that make you better at doing it, right? And one potential answer is it could be that in M1, you're sharpening these cortical inputs with other regions involved in the motor plan, and that's enhancing the motor output. And then in experiment 2, we looked at motor sequence learning to see how what's involved in this kind of rapid consolidation of the motor plan or of the improvement in behavior. And it could be due to these sharpened inputs to the superficial layers of M1 from the rest of the regions involved in the motor processing. And that could result in this rapid offline consolidation. So that answers our second objective question: what can we learn from layer fMRI? That was just two examples of how we've used it to look at the motor cortex. So I'll wrap up pretty briefly here with a discussion of how we might be able to apply this kind of stuff with the work we do here in neurorehabilitation. And it'll be rather brief because I'm still learning and sorting out how this works, right? And I'd be really happy to hear what other people think too about how we can use this. So I've talked to you all about primary motor cortex and the input-output circuitry of across layers in primary motor cortex. It's a pretty basic 2-compartment model. But this the cortex across the entire brain has 6 layers, and they're functionally relevant and differentially organized depending on where you go. So for example, an early visual cortex is a 3-compartment model where inputs from visual inputs from the thalamus, the LGN, go into middle layers, and feedback from downstream visual regions come back to the superficial and deep layers. So understanding, looking across cortex and understanding these input outputs to these regions are going to add a ton to how we understand how the brain works. When we think about these large-scale brain networks that are involved in higher order cognition and perception, memory, attention, all these functions, we tend to think about them as kind of constrained by method, non-directionally connected, or as hierarchies. And the answer, and the real answer to that is much more complex. These hierarchies are recursive. These connections are going forward and backward every step you go recursively. So using layer fMRI is going to get us much further in understanding how these networks are organized and how nodes in them or brain regions are talking to one another and influencing activation across the networks or within them. So yeah, that's basically an illustration of what I just said. So as far as how we might, in addition to just basic science, basic neuroscience and understanding the human brain and its functional organization, how we might apply this to neurorehabilitation, I've always thought about it before coming here as using patients as case studies, more like case studies to give me causal evidence to my brain, to my cognitive or brain models. So someone with a lesion should have some predicted deficit or atypical brain function. But a couple of points to think about potentially for using it in the opposite direction of learning about the laminar circuitry in humans and applying it to rehabilitation. I think first, lesion location is going to affect up and downstream cortical layers differently, right? So understanding where a lesion is can help us to understand our feed forward or feedback inputs being affected more to a given region, and we could build better cognitive hypotheses based on that potentially. So we could have maybe guide therapy by helping us to focus more on a cognitive or a behavioral strategy potentially. And it could also guide localization for brain. So potentially with TMS, do you want to zap upstream or downstream to potentially stimulate spared tissue? Again, I'm new on the job, and I would love to talk more about this, about applying my basic research in cognitive science and fMRI to neurorehabilitation. And so with that, I'd like to thank all of you at Moss and Jefferson. I absolutely love being here. And I'm really excited about growing my lab. If you haven't met Sungmoo and Lori, please say hi to them.
Hi.
They're great people. And I'd also like to thank everybody at the NIH, where I'm coming from, that were collaborators on these two experiments that I described. And most of this work was supported by a K99 and R00 grant from the National Eye Institute. And so I'll take questions, and thanks so much.